Sure. Yeah. So this is an mRNA engineered CAR T therapy for treatment of patients with newly diagnosed multiple myeloma. These cells are engineered with mRNA, as are all of our cells that are in clinical trials and also in development. The reason we focus on mRNA engineering is because we're out to create potent yet safer cell therapies so that they can be used not just in advanced cancers, but also in frontline cancers, as well as even beyond oncology, in our case, in autoimmune disease and respiratory disease. This current program, Descartes 11, is an mRNA engineered CAR T that is targeting a very popular ubiquitous target called B cell maturation antigen. This was originally developed with some technology. It was generated from the NIH and has now been transferred over to Cartesian, supplemented with our existing technology to be able to generate tens of billions of cells in a given production run. And so the cells are frozen, aliquoted, and then administered through repeat dosing within the context of a frontline treatment regimen. The overall design is to improve the percentage of patients that are entering a deep remission, a molecular remission, or minimal residual disease negativity at the end of treatment. Often the problem with multiple myeloma, as it is with many cancers, in fact, many other diseases, is that people take treatments, innovative treatments, are often administered at the very end stages of disease. And it's always easier to treat a disease when it's newly diagnosed or earlier in the treatment paradigm. And it's certainly true with multiple myeloma, because every time the disease relapses, the remissions that follow become shorter and shorter. So multiple myeloma has really benefited over the last couple of decades from an enormous number of innovative therapies that often percolate into a frontline setting in one shape, way, or form at some point, right? And the ability to treat a patient with differentiated therapies in the beginning has allowed these remissions to last longer and longer. Unfortunately, it's still considered an incurable disease. So what we've tried to do is to think of ways in which to bring cell therapy and integrate it directly into a frontline setting, to allow it to be able to integrate seamlessly within the existing standard of care paradigm. And so this particular product, given the fact that it's mRNA engineered, allows us to be able to do that for a couple of reasons. Number one, it's safer, so it can actually be used. It doesn't lead to the kinds of side effects that are now synonymous with CAR T therapy. So in our safety data set, in relapse refractory patients, we have not seen any evidence of cytokine release syndrome, any evidence of neurotoxicity. So it's a safer product. Number two, because we're repeat dosing and administering the full dose of every single dose, we're not in need of clearing the marrow to allow the cells to proliferate into it, similar to how one would do it with conventional DNA engineered CAR T therapies. And so there's no need to administer a preconditioning chemotherapy or a lymphodepleting chemotherapy, which in and of itself can be toxic. That toxicity is often well tolerated, and in a relapse refractory setting, because the risk-benefit ratio at that very advanced stage of the cancer requires that kind of treatment to be available. But in the frontline setting, we believe that a lymphodepleting chemotherapy especially given the fact that the patient is feeling relatively healthy and has other combination therapies that are available to them, is unlikely to be tolerated or preferred. And so we wanted to ensure that when we develop this product, that it could be used in the context of other combination therapies in the absence of lymphodepleting chemotherapy. And the protocol design is actually quite unique because it allows us to be able to go in and out between the end of induction therapy and the beginning of a patient is eligible for it, a stem cell transplant. So upon the completion of induction therapy after several cycles, we then go in and administer the product once every several days over the course of 3 weeks, stop, and then ask, well, have we achieved a molecular remission at week 4? And whatever the outcome is, the hope and expectation is that the patient is going to be better prepared going into later lines of therapy and a stem cell transplant and beyond. And the whole goal is focusing on MRD negativity because epidemiological data suggests that patients who achieve MRD negativity do much better long-term in their treatment. The other important thing to remember here is that these patients were, at least in this study, who are newly diagnosed have high risk characteristics genetically. And so the percentage of patients that achieve MRD negativity on their own is actually relatively low. And so we're hoping that by improving the percentage of patients that achieve MRD negativity, that we're going to be able to make a significant benefit in the long-term treatment of these patients. We're out to cure it. I mean, when we first began this adventure, it was born out of really a personal desire, because right around the time the company was formed in 2016, both family members of mine as well as my co-founders were diagnosed with multiple myeloma. So this is the disease that really originated the genesis of this company. And so when we set out to do something about it, we went and looked at the most potent technology available at that time, which was CAR-T. I still believe it is CAR-T. And yet we recognized that just given the side effects, it just was not prime time for it to be able to be used in a frontline setting. It couldn't be used in a frontline setting because of all the issues that we talked about, the lymphodepleting chemotherapy, the potential for significant adverse effects, et cetera. And so we took that technology and tried to make it potent yet safer to be able to administer in a frontline setting where we can actually eradicate every last clone potentially in the context of combination therapy, not by itself, in the context of combination therapy. I mean, I will just say that we're one of many companies that are working tirelessly in this area, and there's some incredible technologies coming down the road. Cell therapy, I believe, is one of these technologies. Our hope and expectation is that cell therapy, innovation around cell therapy, and whether it's mRNA through our approach or other approaches, enables it to break out of its exclusive or almost exclusive use in advanced cancers. And if we can achieve that through some innovation, we can bring it to frontline cancers, we can bring it outside of oncology into autoimmune diseases, neurology, cardiovascular diseases, respiratory diseases. This is certainly our focus. And I do believe over the next several years, you're going to see a lot of activity beyond oncology and for cell therapy.